Hello, this is Lisa Seidman, and this is the second in a series of videos intended for biotechnology students that are on the topic of amount and concentration calculations. In video 1, I said that there are various ways to express concentration and that I would introduce them in video 2. But instead, I'm inserting a short video that further introduces some concentration calculations that are particularly relevant for biotechnologists. But before we go to the lab, let's stop for a moment at a coffee shop. So here we are at the coffee shop, and let's suppose that I decide I am going to buy a latte. And to make the numbers simple, I'm going to say that it costs $5 for a latte. But a couple of my friends walk in. So now there's 3 of us, and I decide, okay, I'll buy 3 lattes. Then the question is, how much is that going to cost? So this is a how much question, which is actually the subject of this whole video, how much. So you might intuitively know the answer to this. If I want 3 lattes, each costs $5, it's going to cost me $15 for 3 lattes. So this intuitive strategy is probably perfectly fine in our coffee shop at the moment. But sometimes problems are a little bit trickier, and so it's good to have some math strategies that we can apply. So if I know that it costs $5 for 1 latte, and I want 3 lattes, I can multiply by 3. And note that the lattes cancel, and it gives me, again, the $15. And I'm going to call this a unit canceling method. We noticed that the units cancel, and that was good. But there's a third strategy I want to show you, which is called proportions, where we use a proportion equation to solve the problem. So in this case, if it costs $5 for 1 latte, we remember that if we have proportions, we have an equal sign and then something over something. Something is to something as something else is to something else. With proportion equations, whatever is in the denominator needs to have the same units. Whatever is in the 2 numerators has to have the same units. I know that I want 3 lattes, so lattes will have to go into the denominator here. And we're going to put question mark, or the unknown, here. Now I can cross multiply. So I can say $5 times 3 lattes equals question mark times 1 latte. Again, lattes will cancel. I have 5 times 3 is $15. Note the units are dollars, equals question mark. And again, I have this third method, which gave me the same answer as the other 2. Now, if you had proportions way back when and you don't quite remember exactly how to set those up, either you could ask your instructor perhaps for some resources. I would also suggest SolCon videos. These are easily available online, and they are quite good, and there are some that discuss proportions. Okay, now let's move out of the coffee shop and into the lab with some definitions. First of all, a solution is a homogeneous mixture in which one or more substances are dissolved. The solute or solutes are the substances which are dissolved in the solution. And finally, a third definition would be the solvent, what the solutes are dissolved in. Now, we've been talking about solutions in these 2 videos all along. So for example, if we talk about 10 milligrams sodium chloride or table salt per mil, this is a solution in which sodium chloride is the solute. The solvent is almost certainly water, or, because we're biologists, or something derived from water. All right, now let's move along and take a look at a solution. So here's a solution. I'll tell you that the volume is 5 mils, and it contains 10 mgs per 1 mil of solute. In this case, don't try to count the stars. They don't indicate how many. They just tell us that there's something dissolved there. And we're going to ask a how much question now. Although it's going to look like how many. How many or how much? And the question will be how many milligrams are there total in this entire tube? Now, this is another case where you might intuitively know the answer. If there's 10 milligrams in 1 mil, and we have 5 mils, then intuitively we could say, oh yeah, there must be 50 milligrams. But we could also use our math tools. If we could say there's 10 milligrams per 1 mil, we can multiply that by 5 mils. Again, the mils will cancel, and we get 50 milligrams. So this would be our unit canceling method. I'll just say unit C method. Or we could use proportions. If there are 10 milligrams per mil, put our equal sign in our line. Now we note that mils has to go into the denominator. So we'll say 5 mils goes here. We put question mark here. We cross multiply, we get 10 milligrams times 5 mils equals question mark times 1 mil. Mils cancel, and again we get 50 milligrams equals question mark. All right, now just a small note on the next picture. Sometimes we take something out of a tube, a certain volume, and we put it into another tube, and I'll show you an example of that in a few minutes. So in this case, if we take 1 mil of whatever this is out, we knew that it was at 10 milligrams per mil. It's the same as before. 10 milligrams for whatever solute per mil. We take out 1 mil. Now we know that we'll have 10 milligrams of whatever this is in this tube. And sometimes that's what we want to know. So let's move into a word problem. With word problems, what's important, first of all, you want to try to figure out the context. What's happening? Well, we have a solution. It has an enzyme in it. So enzyme is the solute at a concentration of 1 mg per mil. And we say, oh, this is a how much question. How much enzyme is present in the entire solution? Well, this should look pretty familiar. Okay, if we have 100 mils at a concentration of 1 mg per mil, intuitively you might be able to say, I have 100 milligrams in the entire tube, in the entire solution. Or we could use maybe our unit canceling method. 1 milligram per 1 mil times 100 mils. Units cancel equals 100 milligrams. Or we could use proportions. 1 milligram per 1 mil equals, well, the question mark is going to be milligrams, and we're going to have 100 mils here. We can cross multiply 1 milligram times 100 mils equals question mark times 1 mil. Mils cancel, and again, we get that question mark equals 100 milligrams. Right. So now, we've been doing some fairly easy problems where it's pretty, you might have been able to do these intuitively. But let's move into something that's maybe a little more complex and a little bit more realistic. You have a vial of oligonucleotides, which are short molecules of DNA. You move it from the freezer. So now it's sitting on ice. And you have a procedure. You're following some protocol procedure that tells you you need 400 nanograms of oligonucleotides. How much should you pipette from the vial? So there's a liquid here that contains the oligonucleotides. You're going to pipette some of it out of it, put it into another tube. One thing you're going to need to be able to do is read that label, and I know you can't on this picture. So let's go to a picture where you can. So here's the picture of the label. And I'll just note, before we get into the problem, that this is a question where you're going to take some out of a tube, put it into another tube. It's analogous, it's not this problem, but it's analogous to this situation. So let's go back to our label. We're going to get information from this label, but there's several numbers on here. There's 3 micrograms over 50 microliters, there's 22,939 base pairs, etc. And then some other label information. Well, this is a concentration. You can see from the form that it's a concentration, 3 micrograms over 50 microliters. We want 400 nanograms, so this is going to be the relevant information. And again, it is a how much question. But the other thing we have to note right away is that we have micrograms as units here and nanograms here. We have to make sure that they match. So either the 3 micrograms needs to be converted to nanograms, or 400 nanograms needs to be converted to micrograms, which is what I'm going to do here, and say that 400 that's a nanograms, equals 0.400 micrograms. Now, to me, the easiest way to understand this is to set it up as a proportion. So I say, well, if 3 micrograms is present in 50 microliters, put my equal sign and my line, what is to what? So what's going to go in the numerator? Well, it's going to have to be something with micrograms, so that's the 0.400 micrograms, and the unknown is the volume that we want. So we can cross multiply 3 micrograms times question mark equals 0.400 micrograms times 50 microliters. If I do the arithmetic, I come up with 3 micrograms times question mark equals, excuse me, the micrograms will cancel, so they've canceled here, equals 20 microliters. Divide both sides by 3, question mark equals approximately 6.7 microliters. That's 6.7 microliters. So you can check the math and you can get that answer, hopefully. But you might say, well, I want to do this by unit canceling. I'm going to point out here that in your chemistry classes, unit canceling might have been called dimensional analysis, which is a kind of fancy term, sounds pretty cool. But I think that unit canceling is a better way to talk about this because it's more descriptive. So if I use unit canceling and I say that I have 3 micrograms over 50 microliters, which is the way it's shown on the label, and I multiply that by what I want, which is 400 nanograms, or excuse me, micrograms here. That's micrograms. I'm going to get some pretty weird units because I'm going to wind up with micrograms times micrograms divided by microliters. So I'm going to wind up with something micrograms squared over something microliters, which is clearly not right. So it's not going to work to set it up like this. In order to fix this, I need to understand that concentrations are ratios. So if I know, for example, that I get, let's talk about something more familiar, I get 30 miles per gallon from a gallon of gas, that's a ratio. It would also be correct to say that that 1 gallon of gas gives me 30 miles. I can flip over ratios because they have the same meaning. So I can flip over 3 micrograms for 50 microliters and say 50 microliters has 3 micrograms of oligonucleotides. Now I can multiply by 0.400 micrograms. Now my units will cancel, aha. So now I have 50 microliters times 0.4. It's again going to give me 20 microliters divided by 3, which is going to be approximately equal to 6.7 microliters. All right, so that was a somewhat harder problem. Now it's your turn. Why don't you try these? You can stop the video and try to solve these, and then in a few seconds I will give you talk about these. All right, so let's go first to A. Take a look at this. Again, what is the situation here? There is a solution. It has a concentration of enzyme. And again, we have a how much question in both A and B, how much. All right, let's do this by proportions. We want 2 grams. We have 0.4 grams in 1 liter. What's going to go in the numerator? Well, that's got to be grams. And the unknown is how much we want. If we cross multiply, we get 0.4 grams times question mark equals 2 grams times 1 liter. Grams will cancel. And if you solve this and do the arithmetic, you will get 5 question mark equals 5 liters. right, let's try B, and let's try the unit canceling method. But first we note that we have milligrams here, and we had grams up above. So we have to convert something into something else. So let's say that 1 milligram is the same thing as 0.001 grams. All right, now we can say if I have I want 0.001 grams times. Now what do I multiply it by? If I put 0.4 grams per liter, if I write it like that, I'm going to get grams squared. I know that's not going to work out as far as the units are concerned. So I say 1 liter has 0.4 grams. Now my grams will nicely cancel for me, and I get 0.001 liters divided by 0.4, which will give me 2.5 times 10 to the minus 3 liters. And I also do know that that is the same thing as it equals 2.5 mils. And I would want to do that. I might want to do that conversion. 2.5 times 10 to the minus 3 liters equals 2.5 mils, because I can relatively easily measure 2.5 mils in the lab using ordinary equipment. So this is the end of Unit 2 of Video 2, and I will see you in Video 3.